This protocol can clearly differentiate the macrophage cell death modes during Mycobacterial infection. By observing multiple embryos in parallel, it greatly increased the probability of capturing the entire macrophage lytic cell death process.
This protocol may also be applied to the observation of cell death and other cellular behavior in similar scenarios involving infection or sterile inflammation.
During the extended live image, it is very important to keep the intensity of the laser as low as possible to avoid photobleaching and toxicity.
After inoculation according to the manuscript, centrifuge the culture at 3000 times G for 10 minutes to collect the Mycobacterium marinum as a pellet. Discard all but 300 microliters of the supernatant and resuspend the pellet. Add 3 milliliters of 7H9 medium with 10% glycerol to further resuspend the pellet, and then sonicate the suspension in a water bath at 100 watts with 15 seconds on and 15 seconds off for a total of 2 minutes to achieve a single-cell homogenate. Transfer the bacterial suspension to a 10 milliliter syringe and pass through a 5-micron filter to remove any bacterial clumps. Using a spectrophotometer, measure the optical density of the suspension and dilute it with 7H9 media containing 10% glycerol to OD600 at 1. Divide the suspension into 10 microliter aliquots and store at -80 degrees Celsius freezer for further use.